Welcome to the second part of the RBC and WBC biochemistry lecture series. In this video, we'll be concentrating on the RBC membrane proteins that maintain its structure. Let me first give a short overview of RBC membrane proteins. Now, such proteins are identified according to their molecular mass. These proteins can be separated by means of sodium dodecyl sulfate polyacrylamide gel electrophoresis, or SDS-PAGE for short. In this procedure, polypeptides with the highest molecular mass migrate slowest to the gel medium. Each protein component would then show up as bands in the gel medium. Band 1 is the heaviest and thus the slowest to migrate. As will be discussed later, band 1 is given the name alpha spectrin. You will notice as well that as we go along this lecture series, some proteins retain the names of their respective band numbers. This slide features a schematic model of the human RBC membrane. The molecular assembly of the major proteins is indicated. Vertical interactions are perpendicular to the plane of the membrane and are represented by the ankyrin and junctional protein complexes that connect the membrane spectrin skeleton to the integral proteins embedded in the lipid bilayer. Horizontal interactions, on the other hand, occur parallel to the plane of the membrane and involve spectrin tetramers and protein 4.1. Now, we can summarize the components of the RBC membrane into three groups: the lipid bilayer itself, the proteins located in this lipid bilayer, and the proteins that maintain the RBC structure. In this video, we will be concentrating on the third group. The erythrocyte integral membrane proteins will be tackled in the next video. Spectrin is the most abundant protein of the RBC cytoskeleton and functions by maintaining cell shape, at the same time making it flexible, particularly when RBCs traverse narrow capillaries. Spectrin is actually a dimer composed of two intertwining chains, alpha spectrin and beta spectrin. Two spectrin dimers in turn self-associate. And as was illustrated in the diagram previously, spectrin heterotetramers are linked to the inner surface of the cell membrane and are bridged to the other spectrin molecules by way of ankyrin, actin, and protein 4.1. The next RBC membrane protein to talk about is ankyrin. If you would refer to the diagram from earlier, you'll see that ankyrin is pyramidal in shape and it binds spectrin to the cell membrane by way of tight binding to the band 3 protein. Ankyrin serves as the link that secures integral membrane proteins to the underlying cytoskeleton. Ankyrins also function by coordinating functionally related membrane transporters and cell adhesion proteins, and they contribute to the expression, specific localization, and overall stability of the integral membrane proteins. Going back to the SDS-PAGE diagram from earlier, you will see that ankyrin corresponds to band 2.1 in the gel matrix. This band is actually sensitive to proteolysis, which in turn would separate band 2.1 to 2.2, 2.3, and 2.6. Now, let's go back to this diagram from earlier and focus your attention on this area where you have the protein 4.1 and actin molecules bound to the tail ends of the spectrin chains. Protein 4.1, as opposed to the spectrin chains, is globular in structure, and it binds tightly to a site next to the domain on the tail end of spectrin where actin binds. As I'll be showing in another diagram, protein 4.1 appears to mediate communication between the outside of the RBC and the cytoskeleton via the integral membrane protein glycophorin. Protein 4.1 has two forms. The 4.1b form is found in young RBCs. Over time, the asparagine residue in the 502 position of the protein 4.1 polypeptide undergoes age-dependent non-enzymatic deamination, with subsequent conversion to aspartic acid, resulting in the formation of the 4.1a form of protein 4.1. Whether or not this has something to do with RBC aging and signaling for the spleen to remove senescent RBCs from circulation, we do not know. Protein 4.1 complexes with spectrin and actin to form the 4.1 spectrin-actin ternary complex. In essence, protein 4.1, spectrin, and actin are major components of the RBC membrane cytoskeleton. The importance of the 4.1 spectrin-actin ternary complex to the structural integrity of the erythrocyte is best shown by two pathological RBC disorders: hereditary spherocytosis, and homozygous mild hereditary elliptocytosis, both of which will be discussed later under clinical correlations. Also shown in this figure associated with the ternary complex is glycophorin C, as was discussed earlier, and erythrocyte P55 scaffolding protein, which will be discussed later. The next cytoskeleton protein to talk about is protein 4.2, also known as pallidin. Its exact role is not elucidated, but it appears to stabilize the link between the RBC cytoskeleton and the lipid bilayer. As you may have already noted in the diagram presented earlier on, protein 4.2 interacts with several other proteins, including the portion of the membrane-associated protein band3 that faces the cytoplasm. Now, hereditary spherocytosis actually has quite a number of underlying causes. In a particular type of hereditary spherocytosis, however, mutations in protein 4.2 gene occur that result in its near total absence. This type of hereditary spherocytosis is relatively common in Japan. Keep in mind, though, that hereditary spherocytosis itself is a rare disorder. Additionally, protein 4.2 deficiency also occurs in association with band3 mutations, probably as a result of abnormal binding of protein 4.2 to the cytoplasmic domain of band3. Next, we have scaffolding protein P55, also known as protein 4.9. It is part of the earlier mentioned ternary complex formed by protein 4.1. As you may have noted in the illustration I presented 2 slides ago, this ternary complex likewise binds to glycophorin C. It contributes to the strengthening of the link between the RBC cytoskeleton and the lipid bilayer. P55 likewise undergoes extensive palmitoylation. Palmitoyl groups come from a 16-carbon saturated fatty acid called palmitic acid. Palmitic acid is found in cell membranes as a component of phospholipids. The palmitoylation experienced by P55 thus reflects its interaction with the membrane bilayer. Of note, palmitic acid is covalently added to membrane proteins through its cysteine as well as serine and threonine amino acid residues. Palmitoyl acyl transferases are the enzymes that catalyze this reaction. The last cytoskeletal protein to discuss before moving on to clinical correlations is actin. Actin corresponds to band 5 in SDS page. It is of the beta type, and they assemble into short F-actin protofilaments that are of precise length. An actin filament has both a pointed and a barbed end, and there are associated proteins bound to either end. And speaking of proteins associated with actin, they include 2 rod-shaped tropomyosin molecules that are bound along the actin filament. We also have 2 tropomodulin molecules that cap the actin filament at their pointed ends. On the other hand, the barbed end of actin is capped by another associated protein called adenosine, which then forms a bridge that connects the spectrin-actin complex to band 3. Lastly, we have dermatin, which functions by bundling the actin filaments together. It also acts as a linker molecule by binding to membrane-associated GLUT1. GLUT1, for those who don't know yet, is short for glucose transporter 1 and is responsible for internalizing glucose molecules from plasma into the RBC for RBC utilization in the glycolytic pathway. We go back to the same illustration to visualize how actin and its associated proteins are situated in space with respect to each other. Let us now discuss erythrocyte disorders due to abnormalities in cytoskeletal proteins. Hereditary spherocytosis, or HS for short, is characterized by spherically shaped erythrocytes on blood film, along with reticulocytosis and splenomegaly. Recall from your histology lectures that reticulocytosis is a condition where there is an increase in the number of reticulocytes, which are immature red blood cells. It is commonly seen in anemia. They are seen on blood films when the bone marrow is highly active in an attempt to replace red blood cell loss, such as in hemolytic anemia or hemorrhage. Now, because of the diminished deformability of spherocytes, they are more likely to get entrapped in the spleen. Splenomegaly occurs as a result of increased clearance and destruction of these abnormally shaped erythrocytes from circulation. HS is seen in all populations, but appears to be especially common in people of Northern European ancestry. In these individuals, HS affects as many as 1 in 2,000 to 1 in 5,000. This is what HS looks like on peripheral blood smear or blood film. Notice the loss of the normal central pallor of the erythrocytes. This is because the biconcave configuration is no longer maintained as the affected RBCs assume a spherical shape. HS is generally due to deficiency in any of the following cytoskeletal proteins, as well as band3, which is an integral membrane protein. These mutations result in weakened vertical membrane interactions, leading to loss of the biconcave configuration of the RBC and an eventual loss of membrane surface area. In some instances, HS is due to a defective interaction between spectrin and protein 4.1 that leads to a defective ternary complex. As discussed earlier, the diminished deformability of spherocytes make them more likely to get entrapped and destroyed in the spleen, resulting in hemolytic anemia. This is generally relieved by surgical removal of the spleen. The next hereditary condition to discuss is hereditary elliptocytosis, or HE for short, which is described by the presence of elliptical erythrocytes on peripheral smear. HE is relatively common in some parts of the world, although the true prevalence of HE is unknown because many mildly affected individuals are likely to remain undiagnosed. Prevalence has been estimated at approximately 1 in 2,000 to 1 in 4,000 worldwide. HE is most common in individuals of African, Mediterranean, or Southeast Asian descent, paralleling the distribution of malaria. In areas such as West Africa, prevalences of HE as high as 1 to 2% have been suggested. Now, this is what HE looks like on peripheral blood smear. Notice the elliptical shape of the affected erythrocytes. Now, unlike with HS, the principal abnormality of HE affects horizontal instead of vertical membrane protein interactions. Genetic abnormalities leading to HE typically involve mutations or deficiencies in alpha spectrin, beta spectrin, protein 4.1, or glycophorin C. Now, because of the loss of horizontal membrane protein interactions, the cytoskeleton is destabilized and is unable to maintain the biconcave disc shape, leading to the elliptical distortion characteristic of the condition. Hereditary pyropoikilocytosis, or HPP for short, is a rare, severe hemolytic anemia characterized by abnormal sensitivity of erythrocytes to heat. HPP, generally the most severe type of HE, is named because the RBC morphology resembles that seen in patients with thermal burns. Individuals with HPP have often complications of hemolysis, including splenomegaly with or without pigment gallstones that may require frequent transfusions with or without splenectomy. Due to the defective spectrin, individuals with HPP have markedly abnormal RBC morphology, including poikilocytosis, spherocytosis, fragmentation, and extreme microcytosis with mean corpuscular volumes in the range of 30 to 50 femtoliters. Of note, elliptocytes are uncommon in HPP. Now I'll end this video here. Please proceed to the next video where I discuss the erythrocyte integral membrane proteins.